Good afternoon. Topic 11, photosynthesis. So hopefully everyone paid attention during respiration because we are going to be talking about some of the same things with photosynthesis. We're going to be talking about things like electron carriers and electron transport chains. And so there's a lot of similar principles around photosynthesis compared to respiration. The hard thing about photosynthesis is most people are usually a lot less familiar with it. And it's not too bad. If you learn it, it actually gets interesting. The hard thing is to want to learn it. So hopefully you're going to want to do that for Biology 107 because, well, you're going to need to know it for the midterm. And like respiration, I will try to give you an idea of what is kind of the important essential stuff. Because I know photosynthesis is very complex. And I know even if you look at the textbook, they go into things that we're not going to have time to cover in class. And like I said, lots of things to talk about. So before we actually get into the PowerPoints, I'm going to draw a picture for you. So probably today you're going to need some paper, maybe 3 pieces. If you don't have any, maybe grab some. I have some notes and some diagrams. So first one, I'm going to draw a little diagram here. So you can see that I'm wowing you with my artistic abilities. And so hopefully you like those little flowers and that little animal being a bumblebee. So what I want to do is kind of just draw a diagram talking about how these things are related. So let's start off with photosynthesis, and we are going to draw the sun. So there's the sun. There we go. It's very sunny outside today. That's good. Okay, and all right, I think I made it a little bit close. Now I think I can move it. Can I move the flower? How do I do that? Oh no, that's not working. Okay, well, I'll just have to squeeze it in there somewhere. Hopefully it's pretty obvious that what the sun is doing is it's making light. So here we go, here's my light. It's an arrow, there we go, lots of room to squeeze it in. We have light energy, so put that over here, light energy. So this little guy here, by the way, is my autotroph. And by autotroph, I mean something that is self-feeding. So it could be a plant, it could be a bacterium. There's lots of things that do photosynthesis. And let me see here, I'm just trying to use different colors for different things. Maybe I will use the reactants in red. So photosynthesis, of course, takes carbon dioxide, and it's going to take water. So CO2 plus H2O. So you can see we have our inputs of light energy, carbon dioxide and water. And what are we making? Well, we are making organic molecules. So we can write that here. Organic molecules. And this little guy over here is my heterotroph. And I feel like I'm missing something here. Oh, yes, I know what I'm missing. It makes organic molecules, and it also makes oxygen. So that's good because we like oxygen, and so does this little bumblebee. So that is photosynthesis on that side. So maybe I should have titled this photosynthesis and respiration. So what I will do is just make a label here like this. This is photosynthesis. Okay, so on the other side of things, we have our little heterotroph, and our heterotroph is eating food in the form of organic molecules, and he is using oxygen to oxidize those organic molecules. And so what is he making? He's making carbon dioxide and water, but that's not the only thing that the heterotroph is making. The heterotroph is also making, maybe I'll do a double arrow like I did before. So he's also making energy. And I'm just going to write in brackets in the form of ATP. Okay, so one more note, and just enough room to squeeze it on here. Just push that up. Of course, this here is respiration. I think there's a cute little diagram in your textbook showing something like this. I think it's showing a chloroplast and a mitochondria or something like that. So just keep in mind that plants don't just do photosynthesis, right? They also do respiration. So I don't want you to think this is a plant thing and the other thing's an animal thing. Plants also do respiration, but they also do photosynthesis, which is kind of like a bonus for them. Okay, so let's talk about photosynthesis in some detail here. And we're going to get into this and probably just cover the light part, so the photo part today of photosynthesis. And so let's take a look at this. So some things to kind of go over first. We've talked a little bit about autotrophs. And what is an autotroph? It's something that is self-feeding. Sometimes we say photoautotroph, meaning they self-feed by using energy from the sunlight. And like I said, don't just think that we have only plants, right, that do photosynthesis. So if I ask you on a midterm that's coming up in a couple of weeks, who does photosynthesis? Don't just say plants. We've got all sorts of things. You can see we've got, this is some sort of a unicellular protist here. So that's euglena. That's not a plant. We talked about euglena in Lab 5. You have all sorts of algae. This one here is a multicellular algae. Algae is different from plants, by the way. Why is it different? Plants are complex organisms with organs and organ systems and those kind of things. Algae are usually unicellular. Some of the multicellular algae, it's not very complex. There's no vascular systems or anything like that. So it's usually considered different, although obviously very closely related to plants. But not closely related plants, we have cyanobacteria. Those are greenish blue bacteria. And we also have these purple sulfur bacteria. And you can see in the purple sulfur bacteria there, those little granules are actually, they're producing sulfur as part of their photosynthesis process. So maybe I'll mention those a little bit later. So lots of different autotrophs. So where does the photosynthesis occur? Hopefully everybody knows that it occurs in the green part of a plant. And the green is due to the chlorophyll and the chloroplast. So usually that means the leaves. Some plants may have a green stem or other parts, but generally we're talking about the leaves, and other parts of the plant are not, if they're not green, they're probably not doing any photosynthesis. So this is kind of a zoom in on a leaf cross section. We'll kind of come back to this later, and you can see what they're trying to show is the gases going in and out of the leaf so that photosynthesis can occur. So as I mentioned, we did already talk about some chloroplasts in the lab. We were looking at Elodia, and we were looking at the protist euglena. So this week's lab, we're going to be using spinach chloroplasts. And I'm not looking at them under a microscope, but we made a chloroplast suspension for this week's lab. And so you'll learn about that a little bit tomorrow. So our lecture is just in time for the lab. There's another picture. Maybe I showed you this one before. I love these pictures of these organelles that show all the layers, and you can see all the inner workings there of the chloroplast. There's another one there. So what you're looking at, all sorts of membranes in these chloroplasts. So what I want to do for you in a moment here is draw a diagram of the chloroplast and kind of label some of those components. There's another one from a red algal cell. And there's some more pretty cool pictures. One other thing to point out is chloroplasts will store the products of photosynthesis. So in this case, the product being starch, which is a polymer of glucose. So there's the chloroplast that I was going to draw for you. I spent a little too much time on PowerPoint trying to figure out how to draw this. Usually my chloroplast kind of looks like this. We've got two membranes, and then I've got like these guys in the middle. So if you want to draw that, it doesn't have to be as nice. I just want to point out the different components of the chloroplast. So, just like a mitochondria, we have an outer membrane and an inner membrane. So here we go, outer membrane. Bring this closer here. Okay, and then we have an inner membrane. So I'm not going to write a note about the inner membrane and the outer membrane. They're mostly just structural, kind of holding everything in place. They have, there's pores where metabolites can get transported in and out, but mostly, like I said, they're just kind of sitting there holding everything into place. So the next thing to point out is right here, we have another third membrane. So this here is called the thylakoid membrane. I'm not really sure where that word came from. Thylakoid membrane. So I've colored that one green for good reason, because this is the location of chlorophyll. So that will be the note we're going to put. This is the location of chlorophyll. I could write and other pigments if I wanted to, but chlorophyll is the main one. And I'm going to put in brackets, more importantly, this is the location of the light reactions. So that's what we're going to talk about today, light reactions. Okay, and then inside those things, those little flat sort of, I don't know what you want to call them, they sort of look like pancakes or pucks. These are called thylakoids. And the innermost part, oops, how did that erase? Let's try that again. Innermost part is called the thylakoid space. Thylakoid space. So what goes on here? This is where we are going to have a proton motive force. So that's where the hydrogens are pumped. So I can write that if you like, where the hydrogens are pumped. Okay, just enough room on the slide for one more place. Right here, that big open space there, that is called the stroma. So what goes on in the stroma? We've already talked about this a little bit. This is where we have DNA, we have ribosomes. So just kind of, for the functioning of the whole thing, this is where we might have starch granules. And then lastly, I guess what's going to come up today, let's see here. Oh, it's not letting me write on this. Let me try, let me see if I can, it's got to be some place I guess I'll just squeeze it up here. So this is the location of the Calvin cycle. So the Calvin cycle we sometimes call carbon fixation. So sorry about just squeezing everything in there. I thought I had lots of space, but it looks like I can't quite go down to the bottom. It's trying to get me to do something else there. So we've got the chloroplast, we've got all these components of it, and today we want to really focus in on those thylakoids because we want to talk about the light reactions. So let's do that. There's a picture. This is not the one from the textbook, but the one from the textbook you'll see is very similar to what I had, but maybe a little nicer. Has some of the same components labeled. This one here, you can see it's got a stack of thylakoids called the granum. And this is a really neat picture. Again, you've got the electron micrograph on the left and the cartoon on the right. And you can see it's just kind of giving a little bit different detail than what I gave you. So the stroma, like I said, is the fluid kind of around everything. That's where the Calvin cycle occurs, and the thylakoids are those green things, those green stacked pancake-like things where the light reactions are going to occur. So I had mentioned way before, I think somewhere in my notes, this word here popped up, plastid. So I did mention that a plastid, that word would come up again, and at the time I said it basically means chloroplast. But here's the thing, it doesn't always mean chloroplast. Sometimes you see this in scientific literature, people say plastid, and often they mean chloroplast, but what a plastid really is, so plastid is a pigment-containing organelle. So pigment containing organelle. So in a plant, usually that means a chloroplast, right? I mean, most plant cells have chloroplast, and that's the pigment they have. There are other exceptions. Here's a couple here. You can see this one on the left is called an amyloplast. So maybe sometimes you've seen potatoes, and you know, they sometimes have a little bit of color to them. And I'm not sure what the pigment is in there, but it can make them various colors of purple and things like that, or these amyloplasts. And if you think about a plant, sometimes there are other parts of the plants that are colored. So all the ladies are probably thinking, yes, of course there are. Flowers are so beautiful. Of course they are. And that's another plastid for you. Are these chromoplasts that contain all sorts of different colorful pigments that make the flowers really lovely. So if you see that word plastid, usually it's safe to assume that you mean chloroplast, but sometimes it might mean something else. So let's take a look at the chemistry of photosynthesis, and I want to talk a little bit about the components before we get into the light reactions. So if you take a look at the overall reaction, we have carbon dioxide, water, energy in the form of light, and we are going to produce apparently glucose, oxygen, and water. So, you know, sometimes when people are coming out learning this for the first time in like grade 4, they say things like, it's the backwards of respiration. And yeah, kind of, but not quite. The whole thing that's going on here is actually more complex than respiration. And the mechanisms and all that are entirely different. But in some ways, it is respiration reverse in that we are taking carbon dioxide and we're building it up into organic molecules. So let's just talk a little bit about these reactants for a minute. And then, like I said, we're going to talk about the light reactions. So light, of course, we're all familiar with it unless we can't see, is you know, coming from the sun, or sometimes there are other sources, artificial sources for plants. And plants do a very good job at absorbing the visible part of the electromagnetic spectrum. So this is the electromagnetic spectrum. Maybe you're familiar with some of these different types of radiation. You can see we've got gamma rays, X-rays, and UV. Those are pretty high energy things that can give you cancer or sunburns. Visible light is somewhere in the middle. And then you've got on the right all those lower energy radiation, so microwaves and cell phone radiation, things that aren't going to give you a sunburn. And so in the middle is the visible light. So you can see the blue and violet light on the left here is slightly higher energy than the red on the right, and green is somewhere in the middle. So I want to show you, this was a really cool experiment done. I can't even remember what year this was. It was quite some time ago, where people were starting to investigate photosynthesis. And so the question is, well, okay, does photosynthesis work better at different colors of light? So what you can see here is, I don't exactly know how you prepare this kind of thing, but you get a glass microscope slide, and they spread a filament of green algae across it. So algae, of course, will do photosynthesis, so that's good. And then they got a prism and separated the light and made a little rainbow on the glass slide, and they put these motile aerobic bacteria there. So the bacteria are aerobic, meaning they like oxygen, they need oxygen, and they're motile, so they move to areas on the glass slide where there is lots of oxygen. So you can see from this kind of experiment, it was pretty obvious that photosynthesis seems to prefer blue and reddish light, and not so much in the middle. So this makes sense because plants are green, so the green light isn't used, and so that's why they look green, because all the other light is absorbed and the green light is what's left over. So there are more than just green pigments in plants. You probably know that, particularly this time of year where you see lots of different colors before the leaves fall off. Up here, the variety of colors is maybe a little more limited than in southern Alberta. But you know, in people's yards, we get all sorts of oranges and reds, and sometimes even purple and burgundies. So lots of different pigments. Chlorophyll breaks down really quickly. That's why we see all these other pigments in the fall time. and they break down a lot more slower. So this is just the plants trying to capture other types of and colors of light other than just the blue and the red. And different organisms have different pigments, of course. So this kind of just a little bit of a segue to mention what we're going to do in this week's lab. In this week's lab, we're going to do basically two experiments. We're going to do an absorbance spectrum and an action spectrum. So an absorbance spectrum is really just that. What we're going to do is I'm going to take some spinach chloroplasts and put them in one of those little cuvette things. I'm going to stick it into the spectrophotometer, and then I'm just going to take readings of how much it absorbs, going from about 400, I think I go all the way up to 680 nanometers. So in that case, I'm doing the whole chloroplast. If I were to extract those pigments one by one, you might actually get some graphs like this. So you can see somebody's done chlorophyll A. Chlorophyll A is kind of a teal, turquoisey color, and chlorophyll B has a slightly different absorption range, a little bit more green than blue. And then there's the carotenoids, which are shifted even more and get a little bit, just a little bit into the edge of the green range. So we're just going to get one curve because we're measuring them all at once. Like I said, we're not extracting all the pigments one by one. And so this will be part A of your lab, is you're going to make a graph that's plotted like this with wavelength on one axis and absorption on the other axis. So the other part of tomorrow's lab is measuring something called an action spectrum. So what does that mean? That means rather than just measuring the absorption of light, measuring actual photosynthesis. So you can do that in a lot of ways. If you think about the products of photosynthesis, you're making oxygen or you're making carbohydrates, you could measure those things. In tomorrow's lab, we're going to do it a little bit more indirectly. And we're not doing a whole spectrum. We're just going to do 3 measurements, one at blue, one at green, and one at red. So take a look at tomorrow's lab to see what's going on there. But the point of this slide is to show you that they do overlap, right? You get the most amount of photosynthesis and the most amount of absorbance in the blue part of the spectrum, and maybe a little bit less in the red part of the spectrum and a lot less in the green part of the spectrum. So there's chlorophyll, and I kind of like this little model of chlorophyll because it kind of looks like almost a tennis racket. And sometimes that's how I think of chlorophyll, is a tennis racket that is trying to not capture tennis balls, but maybe capture photons of light. So there's chlorophyll A and B again, and I'm showing these structures because if you take enough chemistry courses, you'll learn that when you see structures like that, it has something called conjugated double bonds. I'll write that down for you. So conjugated double bonds. So what does that mean? It means if you take a look, I'm looking at chlorophyll B. I'll point these things out. You've got a double bond, then a single bond, double bond, single bond, double bond, single bond, and it's like that all over the place. So if you're taking Chem 101 right now and you're learning about resonance and all that stuff, conjugated double bonds are really good at that. And what does that mean? It means they're really good at interacting with photons. And in fact, a lot of things with conjugated double bonds have colors to them. And so obviously chlorophyll and chlorophyll A and B do, they have color to them. And they're good at capturing light, which is important for photosynthesis. So it turns out these chlorophylls are, the other thing I noticed from these structures, by the way, is they're very hydrophobic. There's a few oxygens in there, but not a lot. It's mostly carbon and hydrogen. So when I think hydrophobic, then often that means it's found somewhere in a membrane. In fact, that's the case. And this is actually showing some membrane proteins and how they're holding on to the pigments in this chloroplast. And so you can see they've got colored, the chlorophylls are green, and the carotenoids are that orangey yellowy color. So they're in that membrane, in the thylakoid membrane, in fact. And sometimes, you know, because we don't just have pigments, we have proteins. Sometimes you hear people call them reaction centers or photosystems, or, you know, people are using other words to indicate that it's not just pigments. So I'll come back to that in a minute. So second reactant is carbon dioxide. So hopefully, you know, like I said, carbon dioxide comes from respiration, also from burning fossil fuels. And so there's usually plenty of that in the atmosphere for plants. And lastly is water. So water, the very little water a plant actually drinks is used for photosynthesis. But it's obviously essential. And why is it essential? Well, for us, we're looking at this big thing here, this oxygen, we like that. But water is going to provide electrons in the form of chemical bonds. So just like respiration, we are moving electrons around, but usually that means we're moving chemical bonds around. And that's where it's going to come from, is water. Okay, so let's take a look at the kind of big synopsis of photosynthesis. So like I said, you can think of photosynthesis as the photo part, the light reactions, and the synthesis part, the Calvin cycle. So let's take a look at these light reactions here. Shown on the left, you can see all the ingredients. We've got light, we've got water, and that's not quite enough, because what are we going to do with that energy? We're going to transfer it to something in the form of chemical bonds. So what chemical bonds are we going to use? We are going to put chemical bonds in ATP and NADPH. So I'm just going to press a button here and show you that. And so you can see that, like I said, we've got ADP and inorganic phosphate. We're going to attach those two together and we're going to make ATP. So just like in the mitochondria, that's going to involve an electron transport chain and some chemiosmosis. And the other thing we have is this electron carrier, NADP+. So you're probably thinking respiration. Wasn't it NAD plus in respiration? Yes, it was. Turns out this thing has an extra phosphate. So it's NADP plus. But other than that, it's almost identical in terms of the molecule. So we're going to throw hydrogen on that. And the hydrogen is not just the atom, but also the electrons that go with it. And those electrons, like I said, are going to come from water. So this is the light reactions of photosynthesis. And so we are capturing energy in these transient high energy molecules, ATP and NADPH. So that's it. Now we can't store those things up forever. They're short-lived molecules. We've got to store that energy a little bit more permanently, and that's where the synthesis part in photosynthesis comes. So the synthesis part is called the Calvin cycle, named after a guy. Not sure why we haven't renamed it yet. That seems to be the trend. But sometimes we call it carbon fixation, meaning we're taking gaseous carbon, so carbon dioxide is a gas, and we are fixing it into organic compounds. We are attaching it to organic compounds. How are we doing that? Well, we just put all this energy here into ATP and NADPH. That's going to go into the Calvin cycle, and we're going to get our sugar molecules in the end of some sort. And these metabolites here, the NADP+, and the ADP and inorganic phosphate are all going to get recycled and they're ready for more light reaction. Okay, so huge thing to say to you right now. If you are going to learn anything or memorize anything about photosynthesis, this is the slide. Okay, this slide is the foundation of these entire 2 lectures. This slide you should know. Okay. You know, I emphasize this very strongly because, you know, I remember last time on the midterm, I had a multiple choice question that said, what are the products of the light reactions? And like a third of the students couldn't not answer that question. And you can get that right from this slide. What are the products? ATP, NADPH, and oxygen. So memorize this slide and you're going to get some marks on the midterm. Okay. Please memorize this slide. You're going to see it pop up again and again over these 2 lectures on photosynthesis. So do know this slide. Can't emphasize that enough. Notice I'm repeating myself. That's important. Okay, so just that note about NADP plus and ADP are recycled. I think I already said that. And we are making sugar from light. So notice it doesn't say glucose. I know everyone wants to think photosynthesis makes glucose. That's true, not always though. Sometimes these organisms are making other sugars, like fructose, for example, or maybe maltose, which is 2 glucoses attached together. So it kind of avoids saying glucose. Okay, so almost ready to look at those light reactions. I just kind of want to talk a little bit about these photons and what is going on with these photons. So if you look at this slide here, they're showing a photon hit chlorophyll, and then the electrons get excited, and then the photons, you know, they go somewhere else, right? And that's kind of the basis behind how they are capturing these things. So it's somewhat similar to, unfortunately, most of you are probably not doing any labs in person. I did this in high school as well, so I don't know whether you had done this kind of thing, but sometimes you stick things in a Bunsen burner and you stick chemicals in there and they glow all sorts of cool colors. And this is kind of what is happening here. Instead of putting heat energy in, I'm putting light energy in, and then the light energy bounces out. So why would we do this? Just have the energy bounce in and bounce out. Well, the whole idea here is we want to have this reaction center. This is the slide I'm looking for. I'll go back to that previous one in a minute. And you can think of this as a whole array of chlorophyll molecules, right? Just a whole bunch of them laid out to try to, you know, capture that sunlight. Now, not all of them are going to be close to the enzymes that are going to take that and bring it to the next step. So it's kind of like playing hot potato. I caught the photon, I pass it to the next one. I catch a photon, I pass it to the next chlorophyll, and so on. So I kind of like this image here. This is from Wikipedia. They kind of make it look like a big funnel. So you can see the light is going in and just basically bouncing from chlorophyll to chlorophyll until eventually it gets to a reaction center where we're going to have some actual chemistry going. This is the picture from the textbook, and you can see they're trying to sort of show the same thing. You've got these photons, they're sort of bouncing back and forth between the chlorophylls, and eventually the electrons are going to get stripped off and sent to the next step. Okay, so we see that term, reaction center, photosystems, they're kind of just saying, you know, this whole thing is one package. Okay, so let's talk about these light reactions. And I think in the notes for this class, I have all this broken down into, what is it, 8 steps, I believe. And unfortunately, this slide, the numbering is off. This is just an older image from a previous edition of the textbook, but the ones I have in the next slides are correct. And then usually what I do is I write some additional notes on the board to kind of, you know, word things in a different way. And because, you know, if I went through this super fast, you wouldn't retain any of it. So I encourage you to write some notes. As you write notes, even if you don't look at them again, you'll retain more than if you just do nothing. So I encourage you to write some notes. But take a look at what we have here. The main point I want to say on this slide is that plants have 2 photosystems. So notice this is just really bad because photosystem 1 is the second photosystem, and photosystem 2 is the first photosystem. How frustrating is that? Now, why did that happen? Well, it turns out that photosystem 1 was actually discovered first. And then when somebody discovered a second photosystem, they're like, okay, this is number 2. And then they kind of realized how they fit together, and the naming, for the most part, hasn't changed. Nowadays, people are trying to say photosystem 700 because photosystem 1 absorbs best at 700 nanometers. And I believe that's chlorophyll B. No, that's chlorophyll A. And then this one here, photosystem 2, is sometimes called photosystem 680 because it absorbs best at 680 nanometers, and that's chlorophyll B. So, you know, and not that the both chlorophylls do mix and match a little bit, but you know, they tend to stick together. Sorry, chlorophyll A's tend to congregate chlorophyll A's and B's with B's and so on. But you got to really zoom in on the membrane to see that. So let's take a look at these steps here, and we're going to start off with photosystem 2. So I'm going to write some notes here, and usually what I do, like I said, I write this on the board, and I call it linear electron flow. So what do I mean by linear electron flow? I mean it's linear in that it has a start and a finish, right? The electrons have a starting place, and they go step 2, 3, 4, all the way to step 8, and they have a finished place. So rather than cyclic, where things are basically returning where they began, this is linear. Okay, so let's write some notes here. So you can see the very first thing has something to do with light. So that's number 1. So number 1 is the light. Just grabbing my pen in a funny way here. So light, of course, it's the chlorophyll. And you can see eventually the energy is transferred to photosystem 680, which is just more chlorophylls, right? But that's kind of really what I was going to write down because it's related to step 2. So eventually what happens is all of those double bonds in chlorophyll, they capture the photons, and some of those double bonds, because they're electrons, actually get stripped off, right? So I'm going to say that. So we're going to say electrons. So by this I mean electrons found in bonds. Okay. Electrons are, so that E thing here, this is a little short form for electron. Hopefully you know that. Electrons are donated to an electron transport chain. Okay, so I don't want to hit next because I know that it'll go to the next slide and you won't see my notes anymore. So I'm going to talk about water for a moment, which is on the next slide. But I'm going to show you where water comes in because I already told you that water is where the electrons come from. So why am I saying chlorophyll now? Well, it turns out water actually replenishes those electrons into chlorophyll. So that's kind of what happens next. So that's step number 3. And like I said, I'll show that on the next slide. So we're going to say water splits. Okay, so this is a chemical reaction. So we've got H2O. And we have, so the oxygen is released. And the 1/2 oxygen is, I'm just balancing the equation. We have 2 protons. And we have 2 electrons. Okay, and so all of these things go their own ways. Okay, so the oxygen, that's a byproduct. It's supposed to be a P. Let me try that again. Byproduct. Okay, and by byproduct, you know, I don't want to say waste product because we use it. It's good stuff. But the plant at the moment has no use for it. So it's a byproduct. It's going to get released. These protons, so these protons are going to get pumped by the electron transport chain. So I'll put that pumped. Sorry if I'm writing small. I'm just trying to squeeze everything in. Pumped by the electron transport chain. And the electrons, okay, this is just squeezing that in there. And these electrons replenish the chlorophyll. So the electrons lost in chlorophyll. So electrons lost in chlorophyll. So I apologize for the teeny tiny writing. Just trying to get everything squished in there. Okay, I'm just going to give you a moment in case you're just trying to write that down. But just warning you, as soon as I hit next, that's all going to vanish. But the next slide is going to show the water splitting. Okay, so at this point, we're done with the water. It's kind of just a side thing where, oh, we just, you know, we just wrecked our chlorophyll. It's lost some electrons. Let's just replenish that. So bring in the water, and the water does that. Oxygen is released, and the electrons are replenished in the chlorophyll. Okay, so there's the water. You can see it splitting off, and there's the little oxygen, the 1/2 O2. Being released, the hydrogen not being used at the moment, not until we get to that electron transport chain. So there's the electron transport chain. So number 4. So what do electron transport chains do? Well, electron transport chains, they pump hydrogen ions. So electron transport chain pumps hydrogens into, and where do these go? I already told you the name of that is the thylakoid space. The thylakoid space. And this generates a proton motive force. Generates PMF. And write that down for you if you want. Proton motive force. Okay. Oh, number 5 is on here, right? So what do we do with the proton motive force? Hopefully by now you're thinking proton motive force. There's 2 things we can do with it. We can spin a bacterial flagella, or we can make ATP. And that's what we're doing. This is not a bacterium. This is a chloroplast. And we are going to use the proton motive force to make ATP. So ATP synthase there we go, uses proton motive force to make ATP. And if I want to make that sentence longer, I could say from the components of ATP, the components being ADP and inorganic phosphate. Okay, so that's not too bad, right? Like we're step 5 out of 8. So kind of the synopsis so far, light comes and the energy from the light, it takes electrons ultimately from water. Those electrons, that energy is used to pump protons. We made some ATP. So at this point, you know, we still have to, the electrons have to go somewhere, okay? So that's important. And the ATP, we're not going to worry about that until we get to the Calvin cycle, which is going to be, you know, Friday's lecture. So at this point, ATP is good. Plants, cells like ATP. So this is great. So let's move on here to the other photosystem, photosystem 1, okay? So at this point, in theory, this could be done, right? In fact, some bacteria just have photosystem 2, and they transfer their electrons to an electron carrier, okay? But in plants, they have 2 photosystems. They're just trying to capture a little extra light, you know, a little bit in the blue-green area, a little bit in the green-yellow area. And so photosystem 1 is capturing sort of the more blue-green light. And so those electrons, they've lost some energy. And so that's actually, we're just going to give them more energy. We have more light coming in. So you can see number 6, I'll write that down. We have number 6. So number 6 is light. It's chlorophyll. At photosystem 1. There we go, Roman numeral we'll use, so it's easy to tell that's a 1. So that's it at this point. The light, you know, where do the electrons come from that, to replenish that chlorophyll? Those electrons come from photosystem 2, from what we all talked about. So ultimately, way back to step number 3, they came from water. Okay, so in this case, just more chlorophyll is being excited. Those electrons are getting stripped off, and the electrons are getting replenished from the earlier photosystem. Okay, so number 7 and 8. So I'll have to find some place to squeeze them on here. So let me see here. Number 7, I'm going to squeeze it up at the top. So there's number 7. Okay, so the electrons. What was I going to say about this? Number 7. See here, the electrons are donated to a second electron transport chain. Okay. And then number 8, I'm going to do at the bottom. And really the bottom line is, where do the electrons end up? So the electrons, they reduce something. So remember what reduce means. Reduce means giving electrons something. So they reduce NADP+, and we get NADPH. And that's the light reactions, okay? So before we move on, a couple more things to say, right? Take a look at this, okay? What do we have as ingredients for the light reactions? We have water, we have light, okay? And you could talk about ingredients for ADP and for NADPH, so NAD+. And bottom line is we're making these things here, ATP and NADPH. We're making some high energy molecules that are going to be used in the Calvin cycle. So that's kind of the whole goal here, is we're just trying to capture that energy from light. And where do you put energy? You put it into chemical bonds. What are chemical bonds made of? They're made of electrons. Where do we get those electrons from? We get those electrons from water. So it may seem kind of complicated, but you know what? It's working for the plants. They're doing very well for themselves. You know, and they're making some good food for us and other things too. So let's, like I said, things are working really well for those plants. Here's another picture from the textbook, kind of trying to, you know, give you an analogy of what's going on here in terms of potential energy, right? You can think of those photons coming in, and they are giving electrons more potential energy. Now, like I said, the electrons are not by themselves; they are in chemical bonds. But you know, technically, if you do your accounting of where the electrons are and your formal charges and things like that, they are swapping from molecule to molecule. So photosystem 2 promotes an electron, goes through that turbine, ATP synthase makes some ATP, the electrons get promoted again, and they end up in high energy bonds, NADPH. Okay, so there's kind of one more snapshot from the textbook trying to show you how things are arranged in the thylakoids. So you can see those reactions again. You got photosystem 2 and photosystem 1. You've got the light going in, the chloroplast. You've got kind of two things you may notice. One is there's the protons. Okay, they're inside the thylakoid space. So this is where we have a proton motive force. Okay. Those protons, they go through ATP synthase and they make ATP. Ultimately, that happens in the stroma, which is good because that's where the Calvin cycle is going to be. And so the Calvin cycle requires these two things, the ATP and the NADPH. And then the Calvin cycle can happen. So just looking at the time, a couple more things to say, and Calvin cycle will be for next lecture, okay? One more thing to say is that I pointed out that we had linear electron flow. And I told you, what does linear mean? It means the electrons start somewhere. So they start at the water. Eventually, they end up on the NADPH. Right? That's what linear electron flow is for. And what is cyclic electron flow? Well, sometimes the cell just wants extra ATP, right? So it can kind of take those electrons, and rather than putting them back on the NADPH, it just cycles them back through. And so when you've got that electron transport chain, all you're doing is using those energy electrons to pump and make more ATP, because we need more ATP for the Calvin cycle than we do for NADPH. I see there's a question. Someone says, when do you see this? This happens all the time. Both reactions, linear and cyclic electron flow, happen all the time. I will say this though, I will not ask you about cyclic electron flow on the midterm, but this is just, I'm throwing it in for completion to let you know, like I said, sometimes it's important to sort of account for things. And we do need extra ATP, or at least the plant does, or the chloroplast. So there's ATP synthase, exact same slide I showed you before. Exact same reaction. The only difference is I have the labels are different. So remember last time up here, oops. Up here, I did not have thylakoid space. Last time I had intermembrane space because we were talking about mitochondria. And I had matrix written here at the bottom. But now this is a chloroplast. So we're looking at the thylakoid space in the stroma. So I'm going to skip this slide for now. And I just wanted to go back to this slide to finish off. And remind you, if you need to go home and memorize one thing about photosynthesis, you should memorize this slide here. So there's another question I see popping up. Let me see what it says. Somebody is saying one important slide where the products are ATP, NADPH, and oxygen. Yes. So this slide here is very important, like I said. And so right now we just covered the light reactions. So we kind of just looked at, okay, this is not writing here. I'll come back to Darth Vader next day. So we just covered this, right? So there we go. So all we have now is the Calvin cycle left. Okay, and the Calvin cycle is going to take these products. So the water is not important, or the oxygen is not important anymore. What's important now is the ATP and the NADPH, and it's going to take carbon dioxide and ultimately make some sort of carbohydrate. So more on Calvin next day and his cycle, and we'll finish up photosynthesis, and probably we'll also get a chance to start the topic on the cell cycle, which is mitosis and meiosis. So I'll see you next day. Hopefully you guys have a wonderful Wednesday. halfway there through the week, so that's not too bad.